All right, welcome back. Okay, please confirm me. Am I audible to everyone? Just please confirm very quickly. Am I audible to everyone? Can anyone please confirm me that I am audible? Okay, that's great. That's great. Thank you so much. I hope every one of you remember in the yesterday class we have started discussing about the process of initiation, the process of the elongation, and we understand that there are three major steps: initiation, elongation, and termination, right? I hope you all people remember actually what happens in the process of the initiation. Every student very quickly answer my question. Tell me, Shaila, tell me, Divya, how many number of ORCs are present in the prokaryotic organisms such as the E. coli? How many number of ORCs are present in the prokaryotic organisms such as the E. coli? Of course, there's no doubt there is only one ORC is present in the prokaryotic organism. I'll just quickly glimpse it the yesterday class. And one ORC generally contain how many number of base pairs? One ORC contain 245 base pairs. I hope you all people remember. So this is what exactly ORC, friends. Correct na? When we magnify the ORC, what I told to all of you, there is a presence of 5 boxes, 1, 2, 3, 4, 5 DNA boxes. We generally call it as the DNA A boxes, and I hope you remember. On the left hand side, there are 3 more boxes are present, friends. What do we call them as? 13 base pair segments. Correct na? And I believe you all people remember this 13 base pair segments are generally called it as what? DUE. DNA unwinding elements. There is no point of getting confused over there. I hope you all people remember. Why do we call it as the theta replication? I have very clearly told to all the students, whether it is a prokaryotic organism or the eukaryotic organism, friends, mostly what kind of a replication that they will generally show, friends? They generally shows the bidirectional replication only. There is no point of getting confused over there. Correct na? So I hope you remember. So one fork is moving in this way and one fork is moving in the opposite direction. And we also studied very clearly that in case of the circular DNA in the E. coli, whenever the bidirectional replication is happening, the DNA of the E. coli look like a Greek letter theta. What, friends? It looks like a Greek letter theta. That is the reason only we call it as theta replication. We called it as the theta replication. I strongly believe you all people remember what is the major protein that will come and attaches to this particular DNA A element. DNA A protein will come into the picture. Later, who will come into the picture? DNA B along with DNA C, along with ATP, and along with a very important point, along with a protein called as a histone unit-like protein. Along with histone unit-like protein. Okay, all these come close together. They starts opening the double-stranded DNA. I believe you all people remember my statements very correctly. This is the last slide that we are all discussing in the yesterday class. I'm requesting everyone, please pay super attention. I have very clearly explained to all the students the difference between pre-primosome complex and primosome complex. I believe you all people remember. What is the exact difference between the pre-primosome and the primosome complex? If you quickly recall my words, except primer, except primer, the entire complex we call it as what? Pre-primosome complex. Now, so what the pre-primosome complex is generally made up of? Pre-primosome complex contain the DNA A, DNA B, DNA C, topoisomerase enzyme, SSBP proteins. All together we call it as the pre-primosome. Point is very clear. When the pre-primosome is getting converted into the primosome, whenever RNA primase enzyme comes into the picture and whenever it add the primer, then friends, pre-primosome obviously converted into what? Primosome complex. No point of getting confused over there, and it is very easy to understand. Now, I am going to repeat the yesterday's concept in 2 minutes. Every student understand. If any one of you listening the class very first time, undoubtedly it is very difficult to all of you. Okay, you don't understand, you don't get a crystal clear idea. But try to watch the video second time, you get a crystal clear idea, because 99% in case of a molecular biology, friends, most of the time we will expect only experimental based questions only. That is the reason only this topic is this concept, this chapter itself is a difficult chapter. Okay, now listen. Every student say yes or no to me. Now, once I add the primer, what is completed? Initiation process is completed. Now, you tell me, friends, what process has to happen now? That is important point. Now, after the initiation, friends, now the process of elongation has to be continued. Who will do the process of elongation? The DNA polymerase enzyme will come and do the process of elongation. Now listen, all of you, use your brain. Now initiation is completed. Now elongation has to happen. After the elongation, termination has to happen. Correct na? Now every student understand. Before completing of, before completion of elongation and termination, the second round of initiation should not start very immediately. I am saying very immediately. Before the completion of the elongation and before completion of the termination, the second round of initiation should not start. Once the elongation is completed, then it can start actually. It can start. Now understand my statements correctly. Now, if I want to inhibit, listen everyone, understand the point correctly. I want to inhibit second round of initiation. How can I inhibit? If I want to inhibit second round of initiation, how can I inhibit? I can't inhibit the entire enzymes activity. That is not possible, because it is not one or two enzymes. There are multiple enzymes are there. So it is extremely difficult to inhibit all the enzyme. Every student say yes or no to me. Instead of inhibiting all the enzymes, everyone say, if I inhibit the ORC directly, or if I block the ORC, can I say that all the enzymes become inactive? Can I say a statement to all of you? Instead of inhibiting all the enzyme activity, if I block only, this particular ORC, all the enzymes become inactive. Yes or no? Of course, yes. So that process only we generally call it as a sequestration. Everyone try to understand, friends. It seems to be very easy but very confused concept this particular regulation is. Listen, correct. Post initiation ORC site. Listen, understand. What is the meaning of a post initiation ORC site? Post initiation means after the initiation. After the initiation, it is very important that we have to block this particular ORC. Now tell me, why I am blocking the ORC? Tell me. I am blocking the ORC. By that, I can prevent interacting with the replication machinery. and this process only we call it as the sequestration process. What do we call, friends? This process is called as what? Sequestration. There is no point of getting confused over there, and it is easy to understand. Now listen to me very carefully. Palindromic GHC. Now what is my goal? My goal is super clear. I want to block the ORC. Now understand, every student. If I want to block the ORC, one thing is very important. First we have to differentiate which is the parent strand, and we have to differentiate which is the daughter strand. Whenever we can differentiate between the parent strand and daughter strand, friends, then only we can block the ORC. Now question is super clear. Now the question is here, how can we differentiate between the parent strand and daughter strand, or light strand and heavy strand? How can I differentiate? What is the mechanism of differentiating this? Every student understand the point. The difference is methylation. Everyone use the brain. I'll explain twice. Just try to understand me correctly. This is parent strands which are shown in the blue color. Correct na? So you can observe, friends, the parent strand, what kind of a palindromic sequence it is having? It is having a GATC. Right, which is the palindromic sequence. I hope every student can see that. On the adenine, this palindromic sequences on the both parent strands got a methylated. Crystal clear? Now you know very well, during the time of a DNA replication, what happened? Two parent strands get separated. There is no doubt. Two parent strands get separated. Whenever the two parent strands get separated, what is actually happening? Whenever the two parent strands get separated, friends, we call it a daughter strand as, parent strand as the methylated strand. Now question is very clear. What do we call daughter strand? That is an important point. Friends, daughter strand called as hemimethylated DNA. Now all people has to ask me, what exactly the meaning of a hemimethylated DNA? Here, understand, friends, here daughter strands also methylated. Daughter strand also having the palindromic sequence, the orange color ones. Even though daughter strands are having the palindromic sequences, but they are not methylated yet. Whenever daughter strands are only in the non-methylated form, we call it as the hemimethylated DNA. So point is crystal clear. We can separate the daughter strand and parent strand with the help of a methylated groups. Only parent strand will be initially methylated. Daughter strand is not methylated. Whenever it is not methylated, I use the word hemimethylated. At that particular time, there is an important membrane bound inhibitor which we can call SECA. You can see that a membrane bound inhibitor called as SECA. It will come and block the ORC. It will come and block the ORC. As a result, friends, second round of initiation never starts very fastly. Now question is very clear. After some time, there is an important enzyme called as DAM. DAM represents a deoxyamino methyltransferase enzyme. DAM represents what? Deoxyamino methyltransferase enzyme. What is the function of this? Adding the methyl group, that's all. It is adding the methyl group to where? It is adding the methyl group to the adenine present on the hemimethylated DNA. So friends, after some time, the hemimethylated DNA, which is generally a daughter strand, that also become the parent strand. Now we cannot differentiate which is the parent strand, which is the daughter strand. That time what happens? The membrane bound inhibitor SECA will be released out. Then the second round of the replication can start very easily. The people who did not understand, I am explaining you very clearly one more time. Just understand what exactly I am explaining to all of you. My question is very clear here. My point is here, I want all the students has to understand how I am going to block the ORC. That is, you have to understand. I can block the ORC by an important inhibitor called as SEC-A. This is very clear. When the SEC-A can block the ORC, that is the first point. The SEC-A can block the ORC whenever the DNA is in the hemimethylated form. This is the statement you all people have to focus. Whenever the DNA is in the hemimethylated form, then only it can block. Otherwise, it never ever block. There is no confusion at all. So now first, parent strands got methylated, daughter strands are hemimethylated. That time SEC-A will come and block the ORC. Second round of initiation will stop. After some time, very important enzyme such as the DAM, deoxyaminomethyl transferase enzyme, that will come and add the methyl group to the palindromic sequences present on the daughter strand also. Hence, daughter strand also get methylated. Then SEC-A never understand which is the daughter strand, which is the parent strand. Then it can leave from that methylated DNA, and the second round of replication will be continued. This is called as regulatory mechanism of prokaryotic DNA replication, called as sequestration. Is it clear, everyone? Anyone having any doubt, you can ask me. Is it clear, everyone? I'm sincerely requesting anyone having any kind of a doubt, you ask me, because molecular biology is always a crucial topic. Molecular biology is always a crucial topic. You have to remember it. Clear, everyone? Any doubt, small doubt also, you have to please ask me. Okay. All right. Now, whatever we understand is the process of initiation. Now, let's focus on the next and very important mechanism, friends. We call it as the elongation. Now, you people know very well what generally happens in the elongation. Tell me. Elongation, which means that synthesizing the daughter strand. That's all. In a simple word. I'll show you wonderful animation also. You get a brilliant idea. Synthesizing the new strand, that is what happens in the elongation. In other words, I can also say that what happens in the elongation, friends, in the elongation, addition of nucleotides will take place, nothing else. Addition of nucleotides will take place in the process of elongation. No point of getting confused over there. So listen. And what enzyme is involved here? The versatile DNA polymerase 3 enzyme. Again and again I am telling to all the students, here there is no two DNA polymerases are coming into the picture. There is only one DNA polymerase enzyme. But we all people studied very clearly that the DNA polymerase 3 enzyme is what? A dimer. I hope you all people remember. So one core unit having alpha epsilon theta, another core unit also having the alpha epsilon theta. You can see the picture, friends. You can see in the picture very clearly one core enzyme associated with one daughter strand, another core enzyme associated with the another daughter strand. No point of confusion. One core enzyme with one daughter strand, another core enzyme with the another daughter strand. Easy to understand this. So now you also know very well, one strand always moves in the 5' to 3' direction. Look into this picture, towards the replication fork. That's the reason we are calling it as the leading strand. No confusion at all. Now, so this is how the replication fork is moving like, but another strand is moving away from the replication fork, which we generally call it as the lagging strand. Anyway, whenever the lagging strand is getting synthesized, they always synthesized in the form of a pieces, and those pieces only we generally call it as Okazaki fragments. Now, I'll explain this in a more elaborative way. Most of the students do not have an idea how exactly the lagging strand can be synthesized. Most of the people are having a confusion. Now, your confusion will be cleared. Just try to understand the concept. Listen, the elongation phase of replication include two distinct but related operations. One is called leading strand synthesis. Another one is called lagging strand synthesis. Every student understand. Several enzymes at the replication fork are required. Among those, the most important versatile enzyme, that is called DNA polymerase 3 dimer. Friends, this is an important statement to be remembered. It is a dimer. Dimer is the one which is synthesizing leading strand also, lagging strand also. Now again, in the set exam or in the net exam also, you will get a direct question in the exam. What is the mechanism? This is important, friends. This is very important. What is the mechanism shown by the DNA polymerase 3? The mechanism shown by the DNA polymerase 3, we generally call it as the looping mechanism. Never get confused, friends. What exactly the meaning of a looping mechanism? I believe you all people remember what I told to all of you. Whether it is a leading strand, whether it is a lagging strand, what is the common point? The common point is that the daughter strand has to be synthesized in the 5' to 3' direction compulsory. It has to be synthesized in the 5' to 3' direction compulsory. There is no point of getting confused over there. All right, now let's try to understand. So this is called looping mechanism. If you understand this diagram, most of your doubts will be very much cleared. Okay, understand the concept, that's all. It's all about your understanding, how depth you understand, how easily you will understand. Can you see this? I am writing here one parent strand is moving in the 3' to 5', no problem at all. And our DNA polymerase enzyme read the parent strand always in the 3' to 5', and it is going to synthesize the daughter strand in 5' to 3' direction, which means towards the replication fork. You can see the blue color arrow, no problem at all. Okay, here everyone is understood the concept very correctly. Now understand the mechanism behind the lagging strand. If you want to understand the mechanism behind the lagging strand, first you have to understand what is the template strand for the lagging strand. Template strand for the lagging strand is also moving in which direction? 5' to 3'. This is parent strand. So here the major problem. And according to our rule of the DNA replication, daughter strand also has to be synthesized in the 5' to 3' only. Then how is it possible? So that is the reason we are following everything reversible. You understand my statement so clearly. So first, one primer we are adding here. This is one primer. Okay. Whenever we add the one primer, immediately which will come into the picture? DNA polymerase 3 comes into the picture, synthesize one Okazaki fragment. You got it? Again, we are adding one more primer. Who will add it? RNA primase enzyme will add it. Again, DNA polymerase 3 will come and add one more Okazaki fragment. In this way, the process will happen continuously. Now, everyone use your brain and understand what exactly I'm talking here, what I'm saying right now. Can you see that? In the picture, you can see that both the fragments, that is the leading strand and lagging strand, both are moving in the same direction here. You see the arrows. You see the arrows. Why it is possible, or how it is possible actually? It is possible because of the looping mechanism. Friends, what exactly the word looping here? Whenever there is a loop got created, listen. Whenever the loop got created, you can observe that the new Okazaki fragment is always moving towards the replication fork. Whenever the loop got created, then once again primer will be added. Whenever the loop got formed, again the synthesis of Okazaki fragment occur. Whenever the loop got removed, primer added. Whenever loop got created, Okazaki fragment will be synthesized. So listen, the synthesis direction of the leading strand is the same as that of the replication fork. No problem here. But you listen what we have written here. Understand the concept correctly. The synthesis direction of the latest to Okazaki fragment. The synthesis direction of the latest to Okazaki fragment is also the same. As that of the replication fork. This is exclusively possible because of the looping mechanism. So by the looping mechanism, I can conclude to all of you that the new Okazaki fragment also moves towards the replication fork, like however the leading strand is moving. Never ever get confused. This is a unique mechanism that I want you people to understand. In the animation also I'll show you, you get a brilliant idea. Okay, let me show you. Now first understand, you observe this picture. If you observe this picture so clearly, now I am going to ask you only one doubt. I'll ask you only one question. I believe you all people will answer my question. Only one question I'll ask you. First observe this picture. This is the picture actually showing where every enzyme is associated with the DNA. You can see that these are all what? Single stranded binding proteins. This is the RNA primer. Can you see that? Very important one. Hexameric helicase, which is having 6 subunits. See, helicase is so big enzyme. And what is the most important every student has to remember? Whichever I am drawing in the black color, this is entirely DNA polymerase enzyme. See how beautifully they represented. What is this, friends? The yellow color one, whatever you can see, is the beta clamp. The green color one, whatever you can see, is the core enzyme. I hope you are getting me correctly. Same. This is the core enzyme and this is the beta clamp. And I believe you all remember, friends, how can we load the beta clamp onto the core enzyme? I believe you all remember. We can load the beta clamp onto the core enzyme with the help of a clamp loader. Can you see this? This is the clamp loader, which is called gamma complex. Whenever all the enzymes are assembled, listen, every student listen, whenever all the enzymes are assembled, the DNA look like this musical instrument. Whenever every single enzyme assembled, DNA exactly look like this particular music instrument. Now my question to every student is, how many beta clamps we will add in the leading strand, how many beta clamps we will add on the lagging strand? Can any one student please give me the answer? Any one of you? Any one of you? How many leading strands we gonna add and how many beta clamps we gonna add in the leading strand? How many beta clamps we gonna add in the lagging strand? Any one of you? No one is answering. Huh? Always remember, friends, in case of a leading strand, all the students listen, in case of a leading strand, we add only one beta clamp. Because in case of a leading strand, we will add only one primer. I believe every student is getting my point. In the leading strand, we are adding only one primer. Hence, one beta clamp is sufficient over there, which increase the processivity of the DNA polymerase, then the process will occur continuously. But when you look into the lagging strand, we require multiple beta clamps. I'm gonna show you. We're gonna use multiple beta clamps. Now you all people have to ask me, sir, why? Because we added one primer first. Then after adding one primer, the DNA polymerase synthesizes one Okazaki fragment. For synthesizing one Okazaki fragment, we use one beta clamp in the lagging strand. Again, primase enzyme will come and add the primer. Again, we are going to synthesize the another Okazaki fragment. Then again, we use the second beta clamp. So that is how, in case of lagging strand, we gonna use the multiple beta clamps. No point of getting confused over there. Now I'm gonna show you a wonderful animation. You people get little bit clear-cut idea about these mechanisms. I'll just show you in a very quick manner. Always see the effective animations that will help us a lot.
Whatever your thing is, we've got the right domain for you. Search the largest select
like these prokaryotes on the fast.
See, this animation is very elaboratively, it will explain us how exactly the ORC look like, how exactly the DNA is getting replicated. Everything you can understand very elaboratively in this. Okay, focus.
Accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotide.
They are just explaining how does exactly the nucleotide look like. You can see in the picture very clearly. Nucleotide contain nitrogenous base, sugar and phosphate. We all read multiple times. Phosphate group is always the backbone. Because of the backbone only DNA is very stronger. And because of the presence of the hydrogen bonds in the middle, the DNA exactly looks like a ladder.
Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar seen and forms bonds. The double helix will have one strand or deoxyribose. The coded in a is a template in the creation
now look into this. How exactly DNA replication is happening?
Of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication.
Look into this, look into this. DNA replication is not at all happening randomly. I want every one of you focus on it. DNA replication always happen at a specific point. That point only we can call the ORC.
Replication will begin at a specific area of.
You see that specific area which was highlighted here? That is what exactly we can call the ORC.
The molecule called the origin of replication. The origin of replication denotes the area of active replication.
Now I want every single student has to understand the point here. Most of the people do not understand what exactly the replication fork and what exactly the bidirectional replication also. Friends, focus on the picture. You can see that replication fork is the point where the active replication is taking place, and you can see that. And what is bidirectional replication? You people can observe one fork is moving in one direction and another fork is moving on the another direction. Correct or not? That's the reason we call it as the bidirectional replication.
Called the replication fork.
Now see this beautiful picture. You will get a wonderful idea about this picture. Now you people understand. I will explain with the arrows. You look into this picture, friends. In the middle, whatever I am showing, that is the gamma complex. Can you see here the purple color one? Whatever I am showing is the core enzyme. The yellow color one, whatever I am showing is the beta clamp. Now you people observe the concept correctly. In terms of the leading strand, only one beta clamp will be present. In terms of the lagging strand, you all people observe how multiple beta clamps are coming into the picture.
To understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed.
I want you to observe how exactly beta clump keep on adding to the lagging strand, but only how one beta clump is there on the leading strand.
Once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers,
can you see that yellow color one? That is the primer.
Have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the three
look into the continuous synthesis. That is the leading strand, only one beta clamp, only one primer. The process is happening continuously.
prime to 5' oriented parent strand,
This
referred
is the parent
to
strand.
as the leading strand. New nucleotides are added to the 3'
Look into that, look into this. How the nucleotides are adding. We all said multiple times, if I want to add the nucleotide, I told you in the first class, we always require the 3' hydroxyl group. Without 3' hydroxyl group, you cannot add the nucleotide. This is how the nucleotides are adding, exactly.
Prime end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the
Can you see that first primase
5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap.
See, that beta clamp got released out. Now again the new beta
As the
clamp
helix is opened further, this process repeats until
Everyone please confirm me, is it clear everyone how exactly the Okazaki fragments are adding? I believe you all people understand very clearly. In case of a leading strand there is only one beta sliding clamp, whereas in case of a lagging strand multiple beta sliding clamps. Please confirm, is it clear? I believe it's clear for everyone. Alright, so listen, a new beta sliding clamp is then positioned at the primer by the clamp loading complex by the DNA polymerase enzyme. Right, only in case of a lagging strand multiple beta clamps can be used. I hope you all can see here. This is the first beta clamp. Then this beta clamp will be released out, and you can see in the picture again the new beta clamp will be added. I believe in the last class also I told to all the students very clearly. What is the point I told you? Beta clamp, that is a very important statement. I have explained it to you very clearly. The loading of the beta clamp, at the same time unloading of the beta clamp. What is the speciality? Strictly ATP dependent. Very strictly ATP dependent. There is no point of getting confused over there, and it is easy to understand. So we got a little clear idea about the, what we can say, that elongation. Now all the students try to understand. Complete elongation is elongation is completed. Then I believe you all remember what primers we have added, friends. We have added the primer which is a RNA primase. You all people know that point very clearly. Now what we have to do now, we have to remove that particular primer. How can we remove that particular primer? So most of the students will think that DNA polymerase III itself will remove the primer. That is absolutely wrong statement. I believe you all remember what is the length of the DNA primase enzyme. I hope you remember. It is 11 plus or minus 1 nucleotide. That's all. So in order to remove this much small primer, we don't use the polymerase III. For that, in the first class only we read the enzyme DNA polymerase 1, or we can also call it as RNase. Both are same. RNase or the DNA polymerase 1 is the enzyme which will come and remove the primer. Friends, not only removing the primer, and the gap also filled with the DNA polymerase 1 itself. So remember my statements. Removal of the primer, at the same time add the nucleotides or fill the gaps. Filling gaps also can be mediated by that only, as well as removal of the nucleotides also mediated by the polymerase 1 only. Then finally ligase, with the help of ATP molecule, ligase will come and seal the nick or seal the gap. So friends, please never get confused. The enzyme ligase also strictly ATP dependent. No point of getting confused over there. Now the next step I want all the students has to understand, we call it as the process of the termination. This is more important, friends, for the exam point of view. How exactly the process of termination occur? And In case of a prokaryotic termination, every student listen, we require one very important protein. What is the name of the protein that is involved in the termination? Every student remember, we called it as the TUS protein, TUS. Very important protein, friends, it is. The full form of the TUS is terminus, and it is easy. Terminus utilizing substrate. Terminus utilizing substrate. That is the name of the protein. No point of getting confused over there, and it is easy to remember. Correct, Vijay Lakshmi. Now let's understand, friends, how exactly the process of termination occurred. I believe you all people remember very clearly. In case of a prokaryotic organism, the DNA is a circular. You can see the point here very clearly. This is the origin, the point at which the DNA replication is starting. So one fork is moving in a anti-clockwise, one fork is moving on a clockwise direction. So if I want to stop the movement of the replication fork, I require some trap. Suppose example, you are driving a car at 140 kilometers per hour. Very speed. Whenever you reach the toll gate, obviously you will stop. Your speed will become what? Your speed will become absolutely zero. Similarly, how much ever speed the fork are moving, whenever they comes to the trap, they will stop immediately. And what is that trap called as? That particular trap, we call it as tercite. Tercite, we also call it as the termination site. No point of getting confused over there. Every student listen. Suppose this is the origin, one direction clockwise fork, another direction anti-clockwise fork. Now listen all the students. Clockwise direction, there are 5 termination sites are present. Anti-clockwise direction also, there is a presence of the 5 termination sites. Clockwise fork, 5 termination sites, anti-clockwise direction also 5 termination sites. There is no point of getting confused over there, and it is very easy to understand. Now listen, what are those termination sites? On one side we can call H, I, E, D, A. Is it really necessary to remember? It's not necessary, don't worry at all. And on other side we can call C, B, F, G, J. So 5 termination sites are one side and 5 termination sites are located on the another side. Now all the students try to understand the point very correctly, how exactly they are going to block. Focus all of you on the picture. Suppose example, this is the fork. Look into this. This is the fork. This is the fork which is moving in which direction? This is the fork moving in the clockwise direction. You see this. Since it is moving in the clockwise direction, look into this, look into this. It can easily cross the termination sites which are present on the same direction. It can move. But friends, whenever the fork comes here, it will stop. Now what is the reason why it will stop? Because these termination sites are in the opposite direction. I'll explain you little bit elaboratively to you. Suppose friends, this is how the fork is moving. How the termination sites are there? Like this termination sites are there. Okay, termination sites are like this. So it can easily cross all the termination sites because they are also in the same direction. But later, termination sites are located like this. They are located like this. Then obviously it will block there. Similarly, if whenever a fork is moving in this direction, whatever the fork are present in the same direction, it can easily cross. But whatever the forks are present in a opposite direction, it will block over there. So this is how, listen, all the students try to understand my point very correctly. Clockwise replication fork will block at the termination site which is present on the anti-clockwise. Similarly, anti-clockwise replication fork will block the termination site present at the clockwise. I'll repeat it for all of you. There is no point of getting confused. So there are 5 termination sites are present on the clockwise, and this is the fork that is coming from the clockwise. So successfully it will cross it. Now where it will block it? It will block on the anti-clockwise. On other hand, there are 5 replication termination sites are present on the anti-clockwise. Now this is the replication fork coming from anti-clockwise. So it can easily cross these 5. But whenever it comes to the clockwise termination site, it will block here. So in simple words, clockwise replication fork block at the anti-clockwise termination site. Anti-clockwise replication fork block at the termination sites present at the clockwise. Simple. That is how the process of termination takes place. No point of getting confused over there. But what I told to all of you just now, but if this particular termination sites wants to be active, what do they require? If the termination sites wants to be super active, it compulsory require a protein, which what do we call protein, friends? TUS protein. We call it as what protein? TUS protein. Now all the students, please look into this picture without having a confusion. This is replication fork, and what is this? This is a TUS protein. Inside this particular TUS proteins only the 5 termination sites are present. So this replication fork easily cross this TUS. Why? Because both are in the same direction. So it is coming. See this? It crossed successfully. But whenever it reached here, immediately what happened? It immediately blocked. Same thing will happen to the fork coming in this direction also. That's why the TUS and TER proteins are blocking all the termination sites in both directions. No point of getting confused here. This is called as a prokaryotic DNA replication, which we read the process of initiation, the process of elongation, as well as the process of the termination also we understand elaboratively. And do we get the questions really in the exam? Of course, we will get the questions undoubtedly here. Okay. Now the point is very clear. 90% what they will do, 90% of the cases, they will compare prokaryotic DNA replication with the eukaryotic DNA replication. Then they will raise the question. This is a very important to be noted. Now listen. And I can say to all of you, most of the time questions comes from the eukaryotes. I'm not at all saying, friends, questions never come from prokaryotes. Questions do come from prokaryotes. But compared to the prokaryotes, the most probability of asking the questions are from the eukaryotes only, because the genome is complex. Mechanism is also very complex. Enzymes involved is also more. That's why they arise more experimental based questions from the eukaryotes only. Alright, now let's try to understand the concept correctly. The DNA molecules in the eukaryotic cells considerably larger. Of course, the size of the DNA is very larger compared to the prokaryotes, and most importantly, they are organized into a very complex structures. They are organized into extremely complex structures. No point of getting confused over there. Now, I believe every student remember my statement. In case of a prokaryotic organism, how many ORCs are there? First understand the differences. In prokaryotes, there is only one ORC. Correct, na? In the eukaryote, we don't use the word ORC. I believe you remember. In the eukaryotes, instead of using the word ORC, we use the word called as a replicator. We use the word called as what? Replicator. No point of getting confused over there. And how many replicators are there? Nearly, I am talking nearly 400 replicators are present. So imagine how big the eukaryotic genomes are. However, while studying the prokaryotic organism, we always take E. coli as the best example. Similarly, friends, whenever we are studying the eukaryotic organism, we always take yeast as the best example. Correct, correct. In case of a yeast, the replicator having the other name. What is the name of the replicator in the yeast? We call it as the ARS, autonomously replicating sequence, which is present in the yeast. No point of confusing, friends. So always compare and understand. You remember, in case of a prokaryotic organism, ORC made up of 245 base pairs. What does it made up of? Tell me. It is made up of DNA A boxes, and it is also made up of DNA unwinding elements. I believe you all remember. Similarly, friends, if you look into the ARS in the yeast, it is made up of A, B1, B2 and B3. A, B1, B2 and B3. No point of getting confused over there. Now, let's try to understand the most important and unique point, because this is where we're going to get the questions in the exam. We already got the question that is exclusively from the regulation, that too in the graphical form, and we will understand elaboratively, but you focus first. In case of a eukaryotic organism, the DNA replication is always related with what? Always related with this particular cell cycle. You all people know very well, in the cell cycle we have G1 phase, we have S phase, we have a G2 phase, and we have a M phase. So remember my statements very correctly. What we always say that? We always say that G1 phase is what? We always say that G1 phase is what kind of a phase? G1 phase is the preparatory phase. Correct or not? G1 phase is the preparatory phase for the S phase. And what is going to happen in the S phase? DNA replication is happening. Similarly, however, we can call G1 phase is the preparatory phase for the S phase. Similarly, friends, we can call G2 phase is the preparatory phase for the M phase. What do we call? G2 phase is the preparatory phase for the M phase. There is no point of getting confused over there. But anyway, we all people has to focus majorly on the G1 phase and S phase. Most of the students will get confusion here. People think that DNA is preparing itself in the S phase and DNA is replicated also in the S phase. Now you listen my statements. 100% of preparation. I'll explain you elaboratively in the upcoming slide, but you first remember 100% of preparation happens in the G1 phase. Now you can ask me, sir, what is exactly the meaning of a preparation? Which means that all enzymes are getting ready. Helicase is getting ready, topoisomerase is getting ready, SSBPs are getting ready, primase is getting ready. Every enzyme will get ready in the G1 phase. Then all the students has to get a wonderful doubt. Sir, if all the enzymes are ready in the G1 phase, then why the DNA replication is not at all happening in the G1 phase? That's a wonderful question. Why? If all the enzymes are getting ready in the G1 phase only, why the DNA replication is not happening in the G1 phase? The point is very clear. They do not have a license. They do not have a license to start the DNA replication in the G1 phase. They will get the license only in the S phase. That's why the cell enter into the S phase. Then only the cell undergo the DNA replication, that too in the S phase. I will tell you in the upcoming slide elaboratively. No point of getting confused. And keep answering my questions. It is always confusing, friends. So listen. Let's start with the initiation. Same phases. Whether it is a prokaryotic replication or eukaryotic replication, the three processes are same: initiation, elongation, termination, but in a little different way. When you consider the initiation, all the students listen. I told you, right? It is happening in two phases. One is called G1 phase in the S phase. Friends, don't get confused. I am talking only initiation. The process of initiation of DNA replication in eukaryotes takes place in two phases, G1 phase in the S phase. In the G1 phase, pre-replicative complex will form. Now you have to ask me what is called Pre-replicative complex means assembly of all the enzymes. All the enzymes got assembled. But what I told you, they did not get the license. When they did not get the license, friends, they never ever become active. But if they want to become active, what they required? The second step is activation. So when they become active, only in the S phase. So friends, pre-replicative complex converted into active initiation complex only in the S phase. That's why they'll always give you confused statements and they'll make you confused. Pre-replicative complex means all the enzymes are ready but they did not get the license. Once they enter into the S phase, origin become activated, pre-replicative complex will convert it into active initiation complex. Then only the DNA replication will starts. Now see here friends, it is a little bit interesting. No point of confusion. I'll show you the diagram. What is the first protein that is involved in the prokaryotic DNA replication? I believe all the students remember DNA. I believe you remember. DNA A is the protein which comes and attaches to the ORC. The best way to remember is the comparison. Now in case of eukaryotes, what is the name of the protein? See friends, don't confuse. Name of the protein only called ORC. Origin recognition complex will come and attaches to the replicator. Friends, never ever get confused. Name of the protein only called as ORC, origin recognition complex, comes and attaches to the replicator. So first ORC attaches, I'll show you the diagram. Then ORC recruit CDC6. CDC6 will come and attaches to the ORC in a ATP dependent manner. This they will give in the exam as one of the statement. Then CDC6 and ORC recruit CDT1. Then total how many proteins are there? ORC is there, CDC6. Sometimes instead of CDC6, 18 will come. And CDT1. Then finally, ORC, CDC6 are 18, CDT1, finally recruit MCM. What is MCM? MCM nothing but helicase. And where all the events are happening? All the events are only happening in the G1 phase. Look into the picture, you get a crystal clear idea. Look into this. This is the protein named as ORC protein. Where exactly this protein will come and attaches to the replicator. Correct na? Then ORC recruit what? Either CDC6 or 18. Then after CDC6 or 18, CDT1 come into the picture. Then CDC6, 18 all together recruit MCM. Can you see that? MCM is what MCM actually? It is a helicase, hexamer, having generally 6 subunits. So till here friends, we called it as a pre-replicative complex. But the pre-replicative complex is not at all active. When it will become active? It will become active only in the S phase. Alright. Is it clear every student till this particular slide? In the next class, we will complete this entire eukaryotic DNA replication. I am sincerely requesting, I'll post a complete PPT in the group, compulsory revise. Just keep the PPT with you once and read the study material once, compulsory. Then only you can answer the question. The questions are very difficult from the molecular biology. Let me solve you the paper. Clear everyone? Anyone having any doubts, you can ask. Okay. That's all for now. Thank you all. I'll see you in the next class. Okay. Thank you all. Keep reading. Do not waste the time. Okay. Thank you all. Thank you.